Hello, people. Welcome back. Happy New Year. I think we're all filing in. Cool. Give everybody just a second. Nope, too fast. Not fast enough. All righty, well, we'll go ahead and start since most of you guys are in here already. Hope you had a wonderful Christmas. Happy New Year. So today we're talking about viruses and prokaryotes and how we get from our most basic life forms to plants. So we're kind of moving from least complex to more complex in this quarter. And then our final quarter will be kind of more human body focused. So viruses, what are viruses? A virus is an infectious agent that can only replicate within a host organism. Viruses can infect a variety of living organisms, including bacteria, plants, and animals. Viruses are so small that a high-powered microscope is necessary to visualize them, and they have a very simple structure. When a virus particle is independent from its hosts, it consists of its viral genome, or genetic material, contained within a protein shell called a capsid. And in some cases, that protein shell is enclosed in a membrane that they also call an envelope. So viral genomes can be very diverse, since they can be either DNA or RNA. They can be single or double-stranded. They can be linear or circular, and they can vary in length and the number of DNA or RNA molecules and sequences involved. So the thing to remember about viruses is that when we're referring to them, you don't want to use the term like viral cell because that's not a thing. And you don't want to use the term viral molecule. I've seen a lot of that lately with COVID, people saying stuff. Particle would be the more appropriate term if you're going to use a term. And we also have to remember they're not living things. They are an infectious agent, but they're not alive. And we're going to get to that in just a minute. So discovery of viruses. So let's talk a little bit about kind of what we know about basic structure and how we get to discovering that basic structure, sorry. So a viral particle is made of your nucleic acid, so your DNA or RNA, protein, and in some cases lipids. Remember that lipids are fats and oils, and that so in this case what we've got here is a coronavirus. So this is probably pretty familiar to most of us by now, right? So here you have inside, this is your RNA viral genome, which is going to mean that your coronavirus is an RNA virus, and then it's going to have your membrane and your capsid. Basically, this is what contains this is what contains your viral genome. And the outside are these things they call spikes. And we will get back to this in a little bit. So I want you to kind of commit this image to memory, because when we start talking about how our immune system reacts to viruses, these little guys are going to be very important. All right, now they also can differ widely in size and structure, and are so small that you can only see them with an electron microscope. Even like my best microscopes that I have, and you know that you guys all have some here, and then I have one that's even more high powered, those won't see viruses. You're going to have to get an electron microscope to see a virus. So your typical virus has a core right here of DNA or RNA surrounded by a protein coat called a capsid. And then sometimes, like with influenza or flu virus, you're going to have a membrane surrounding that capsid. Now, this is important because as we're going to discuss, you can't kill a virus because it's not alive. So in that case, how do you prevent them from spreading? Well, one thing you can do is destroy that protein capsid that encloses the RNA or the DNA, and that basically makes it a non-functional entity at that point. So that's why hand sanitizer works, because the alcohol will denature or destroy that protein and break down basically the exterior of the viral particle, and then it doesn't work anymore. So your simplest viruses are going to contain only a few genes, where the most complex ones are going to have hundreds of genes. And lastly, the type of virus that infects the bacteria is called a bacteriophage. This one is T4. There's a couple ones you may hear referenced in today's lesson, and those are just sort of the ones that are more well known because they're often used in research. And the reason for that would be because if you are trying to study viral behavior or viral replication, it's really good if your subject can be a bacteria and not maybe a more complex living organism. So you can see how your bacteriophage interacts with your bacteria, and that can give you good information on what you can anticipate from bacteria in other types of hosts. And bacteriophages, they tend to look like, they really do look like something NASA came up with. They don't look like something nature created. They're really cool. They have, this big head and a collar and a sheath and all these like long tail fibers. And you can watch YouTube videos where they've basically made graphics of them coming in and landing on cells and injecting their genetic material. It's pretty cool. So who discovered viruses and how were they discovered? So 1892, Dmitry Ivanovsky. So this is a Russian guy. He's a 28-year-old Russian biologist. And he demonstrated, he can do a lot by the time he's 28, y'all. He's obviously not 28 in this picture. But he demonstrated the cause of a particular plant disease called the tobacco mosaic disease was found in the liquid extracted from the plants, but he couldn't pin down the culprit. So 1892, if you can think back then, like turn of the century, Victorian era, really cool fashion at the time, big hip rolls and corsets and fun things. And this dude is extracting juice from these infected tobacco plants, and he was able to discern that whatever was causing this disease to get passed from plant to plant was in the juice, but he didn't know what it was. So about 5 years later, we get this Dutch dude named Martinus. I think it's Bijnerek. I always have trouble with this one. I always have trouble with Dutch names. I was telling the junior group, it's funny, when we were in Amsterdam a couple years ago, we got so used to everything sounding so weird that we made up our own words for stuff. Like when we would leave the hotel, or we wanted to remember to bring our bakken with us so we could buy things and put them in. So anyway, he suggested that there were actually tiny particles in the juice that were causing the disease. And he named them after the Latin word for poison. So he's the guy who coined the term virus. So just around the turn of the century. And then we got to go a little further along in history here to 1935, and we get this guy here, Wendell Stanley. He isolated crystals of the tobacco mosaic virus. And he noticed, and it looked like this, it's pretty cool, but he knows that living things don't crystallize. So he inferred then, based on the knowledge he already knew that living things don't crystallize, that viruses are not alive. So this is the conclusion that all biologists recognize today, is that viruses are non-living particles made of proteins, nucleic acids, and sometimes lipids, but that they are not alive. So viral infections, this is what we're all dealing with these days, right? So there are two main types of viral infections. There's a lytic infection, and this is when a virus enters a bacterial cell, makes copies of itself, and causes the cell to explode, or what we call lyse. And bacteriophage T4, which is what we saw just a minute ago, a little graphic of it, T4 is an example of a bacteriophage that causes such an infection. So it will infect the bacteria and then cause that cell to burst. So it has a DNA core inside a protein capsid. It's going to bind to the surface of its bacterial host. It's going to inject the DNA into the cell, make mRNA from viral genes. And then that viral mRNA is going to be translated into viral proteins and act like a molecular wrecking crew. And it's going to just chop up the cell's DNA. And then once those viral genes are controlling the cell, the host cell now makes thousands of copies of viral nucleic acid and capsid proteins, and it enables the virus to reproduce. Viruses cannot reproduce without a host. You can put bacteria down on a surface, and if conditions are favorable, it will reproduce and grow. But a virus will not reproduce without a host. Now, it can be, I don't, survive isn't the right word, I guess, because it's not alive, but it can exist and still be functional on surfaces, but it can't reproduce. So viral DNA is assembled into new viral particles, and then before long, this infected cell is going to lyse or burst, and all of these hundreds of viral particles are now going to go on to infect other cells. And that is the lytic cycle of infection. The other type of infection is called a lysogenic infection. So in some cases, like in bacteriophage lambda, which is another one they've studied, the host cell is not immediately taken over. So your viral nucleic acid is going to be inserted into the host DNA, and it's going to be replicated along with the host DNA, but it's not going to damage the cell. And here's why this gets crazy, because your viral DNA is going to multiply as your host cell reproduces. So as your host cell divides and produces daughter cells, it's taking the viral DNA with it. So in this way, each generation of daughter cells derived from the original host is infected. The bacteriophage DNA that becomes embedded in the bacterial host's DNA is now called a prophage. So the prophage can remain part of the DNA of the host cell for many, many generations. And influences from the environment, things like radiation, heat, certain chemicals, they can trigger that prophage to become active. And in that case, it's going to remove itself from the host cell and reproduce by forming new viral particles. So this is a much longer phase, this lysogenic phase. Lysogenic phase can last, any amount of time until the right trigger causes the actual production of new viral particles. So the lysogenic infection now becomes lytic. So now you have cells exploding everywhere. And the details of viral infection in eukaryotic cells, remember, those are cells with nuclear bound membranes, differ in many ways from viral infections of bacteria by bacteriophages. But for the most part, the basic patterns of infections in animals and other eukaryotes are similar to lytic and lysogenic infections of bacteria. So we can use lambda and T4 bacteriophages and see how they behave in bacterial cells that they infect. And then we can extrapolate some of that data to eukaryotic cells, so therefore animal and plant cells. So here are our favorite amoeba sisters again, and they're going to kind of summarize some of what we just discussed.
Chances are in your life, you've had the common cold before. It can cause sneezing and coughing and can make you feel awful. The worst thing about the common cold is that you can take medications to help with the symptoms, but you really can't treat the actual cold with antibiotics like you would a strep throat infection or an infected tooth. Why? Well, let's talk about what causes it: a virus. The common cold is caused by a virus. While all living things are made of one or more cells, see the modern cell theory, viruses are not cells. Viruses aren't technically classified as living things since they lack many characteristics of life, but there is some debate in calling them non-living as well. Medications like antibiotics attack bacteria like E. coli, which are living cells. Antifungal medications can be used on fungi like athlete's foot, which are made of living cells. Viruses are not bacteria or fungi. You can't classify a virus as a prokaryote or a eukaryote either because it's not a cell. So what is their structure like? First of all, viruses are much, much smaller than cells. You typically are going to need an electron microscope in order to see a virus. Viruses come in many shapes. Our favorite one to draw is the bacteriophage. It sounds very fancy, but it's a type of virus that tends to attack bacteria. Despite this being our favorite to draw, know that the shape of viruses vary greatly. One thing all viruses have in common is that they have some type of genetic material. This genetic material can be in the form of DNA or RNA. Viruses usually have some kind of protein coat, also known as a capsid. The capsid can protect the DNA or RNA genetic material. Viruses can also have other structures. Some viruses package special enzymes with them. Some viruses have an outer envelope. These additions can be useful to the virus in virus replication. One thing that makes virus replication or reproduction unique is that they can't replicate without a host. And it's their reproducing that tends to be a problem for its host, whether the host is you, in the case of the common cold, or an insect, plant, this bacterium. Viruses tend to be very specific for the host that they're going to infect, but anything that is alive can be a host because cells have the machinery that viruses need in order to replicate. We're going to talk about two types of viral replication cycles. The first one is the lytic cycle. In the lytic cycle, the virus, remember, very selective, attaches to a host cell. It often binds to a receptor that this cell has, which gives it access to attach. The virus can inject either its DNA or RNA, depending on what kind of genetic material it has, into the cell. Some types of viruses are actually taken inside the cell themselves. Now, you would think that the cell would notice viral DNA or RNA or an entire virus that has been taken in, but in many cases, it does not. It takes the genetic material from the virus and it starts following the instructions, which in this case is very bad because the instructions tell it to make copies of the virus. The cell uses its own resources to start building. It starts making so many copies of the virus that it can cause the cell membrane of the host to rupture, explode, lyse. So what happens is now that these new viral copies, they can get out of the cell and go and infect other cells. This is known as the lytic cycle. By the way, the lysing of the cell membrane is a very bad thing for the cell. The cell cannot survive without its cell membrane. The other cycle is called the lysogenic cycle. And I like to remember this is the longer word. So I like to think of this as the longer, sneaky cycle. Viruses that go through this cycle tend to do the same thing at the beginning. They inject their genetic material, but this time the genetic material stays hidden in the host genetic material. What happens is that when the host makes new cells, it replicates its own genetic material and the viral genetic material. Then those daughter cells make new cells, and they also replicate their own genetic material and the viral genetic material, and so on. This is the lysogenic cycle. That may not seem like a problem, but it really becomes one if the virus gets triggered to go into the lytic cycle from that point, because then all those cells that have the viral genetic material can start assembling viruses. And as far as what triggers it to go into the lytic cycle, it can vary. It could be a chemical trigger, for example, or a lack of food for the host. Going back to the common cold, a lot of times it has to run its course. Your immune system will typically take care of it, and medications can help with the symptoms so that they don't feel as bad. But unfortunately, some viruses are extremely complicated, like HIV, a virus that can lead to AIDS. Like most viruses, it is very specific. It binds to a CD4 glycoprotein that is found on the surface of specific cells, such as helper T cells. The problem about this particular target is that helper T cells play an important role in protecting the body and the immune system. Since HIV goes after these immune cells, it can make a person that is infected with HIV vulnerable to other infections. There are medications that have been developed to help keep HIV from replicating as quickly, and there continues to be research into looking into ways to keep the virus from attaching, because if the virus can't attach, it can't insert its genetic material. A problem that continues to be a challenge is that many viruses, including HIV, can mutate. So a treatment developed against a certain virus type may not work on a mutated form. So with our talk about viruses, you might wonder, is there anything positive about viruses? Well, some viruses can play a useful role in gene therapy. Definitely something to explore more. Also remember how we mentioned that viruses can go after other organisms, like insects, for example. A virus that targets pest insects could be an alternative to a chemical pesticide. One such example virus that you may want to explore is the nuclear polyhedrosis virus. Still, it's important to consider that whenever talking about any kind of pesticide, chemical or viral, there could be consequences in the ecosystem. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Do love them. I'm gonna leave this up for a second. So if you want to pause it and then check your answers on that, you can. All right, so prokaryotes. Now, our prokaryotic cells are comprised of both bacteria and archaea. All right, their genetic material isn't stored within a membrane-bound nucleus. Remember, that's what delineates prokaryotes from eukaryotes. It's stored in what they call a nucleoid that floats around in the cell's cytoplasm. Prokaryotic cells are normally smaller than eukaryotic cells, and their typical size can range from 0.1 to 5 micrometers in diameter. And prokaryotes are made up of a single cell, although they can pair up or cluster together to form mats. So how do we classify them? This is always complicated. Next week gets even more complicated when we get into how do we classify protists. But a lot of the times when we start classifying prokaryotes and we break down our bacteria and our archaea, this part's more simple. Next week it gets more complicated. So bacteria are diverse. They range from disease-causing pathogens, we're going to come back to this word, beneficial photosynthesizers and symbionts. All bacteria, except for mycoplasmas, we'll come back to that later too, possess semi-rigid cell walls composed of peptidoglycan. Okay, peptidoglycan. Think for a minute, what else has cell walls? Plants. And what are plant cell walls composed of? Cellulose. There is another type of cell wall, and then we'll talk about those, and those are the ones that are in fungi. And when we get to talking about the fungus and how it functions, you'll find that the cell wall of a fungus is composed of something called chitin. So with 3 different materials that are used to build cell walls. And these are a little less rigid than plant cell walls. So peptidoglycan. So archaea are diverse, but none are pathogenic and many live in extreme environments. So down here you have your prokaryota and then your eukaryota, right? And you got your bacteria over here and your archaea over here. So your archaea are never going to be pathogenic. Okay, so here's the word again. Pathogen is disease causing. We'll come back to that in a minute in a little more detail, but anything that causes disease. So your archaea don't cause disease, but they do live in extreme environments like those. Remember we talked about the deep sea vents or where there's like a lot of sulfur, that sort of thing. So right here, we can move this down here. We can kind of get an overview of them. So your archaea and your bacteria. Your archaea constitute a domain or kingdom of single-celled microorganisms, microbes and prokaryotes, meaning that they have no cell nucleus or any membrane-bound organelles in their cells. Whereas bacteria constitute a large domain of prokaryotic microorganisms, typically a few micrometers in length. They have a number of shapes ranging from spheres, rods, and spirals. We'll see those in a minute. Cell walls, again, we have pseudo peptidoglycan. So this is even more different. This is even more different. This is not even quite the same compound as here. And then there's also a lipopolysaccharide that can exist in bacteria, but typically we're dealing with this peptidoglycan. Habitats, so archaea in extremely harsh environments like hot springs, salt lakes, marshlands, oceans, guts of ruminants in humans. Ruminants are those animals that have two stomachs, like think about cows. Whereas bacteria are ubiquitous. They're found in soil, they're found in radioactive waste, they're found on surfaces, they're found everywhere, okay? Easy places to live and hard places to live. Growth and reproduction. So your archaea reproduce asexually by the process of binary fission, budding, and fragmentation. And we'll talk more about these types of reproduction over the next couple weeks. Eubacteria produce asexually through binary fission, budding, fragmentation, but they have a unique ability to form spores and remain dormant over years as a trait not exhibited by archaebacteria. And then when we get into protists next week, we talk about protists, there are other types of asexual and sexual reproduction in which they all engage. So structure and function, we're going to categorize our bacteria mostly based on their shape. So size and shape. Prokaryotes are probably the smallest living organisms, ranging in size from 0.15 micrometers, which are mycoplasms, to 0.25 micrometers, like chlamydia, to 0.045 micrometers, which is like Rickettsia, and about 2 micrometers, which is many bacteria. So you can see that they're going from, little itty bitty things, 1/4 of a micrometer to 2 micrometers. And then you have certain prokaryotes, such as bacteria, that occur in different forms. So here's how we categorize by shape, less often than size, but by shape. These are great words. You're going to love these guys. So if you have a little ball, a sphere, it's called a coccus. Together they call them cocci. And then if you have a rod, it's a bacillus or bacilli if it's more than one. These little comma-shaped ones are vibrio. And then we get these fun, like kind of wavy, flexible ones. These are spirochete, or some people say spirochete. And then we have corkscrew or spirillum. We've also got some prokaryotes that are sort of vary, and those are pleomorphic. Rickettsia that we talked about before and mycoplasmas are examples of pleomorphic microorganisms. So there's some that just sort of kind of take on amorphous type of shapes. And while when certain prokaryotes divide, they cling to each other, and they'll do it in a very distinctive way. And it's always the same for that particular species. So for example, I love this. A diplococcus is a pair of cocci. You may have heard of a diplodocus, that's a type of dinosaur, but a diplococcus is a pair of cocci. And then you have streptococcus. If you've ever had strep throat, you've had streptococcus. That's a chain of cocci. And then tetracoccus is going to be 4 cocci arranged in a little cube. Aren't these fun? Staphylococcus is when you get them arranged like a cluster of grapes. And then you can also have streptobacilli, for example, and that's like bacilli in a long chain. So your strep would be your chain in this case, because you see that root used here again, not root, but prefix used here twice. So importance of prokaryotes. I put some froyo in here. You'll see why in a second. So prokaryotes are important to all life on Earth for a number of reasons. They play a critical role in recycling of nutrients, decomposing dead organisms, and allowing their nutrients to be reused. They're also important for many metabolic processes. So we've got our decomposers, we've got photosynthetic prokaryotes, such as cyanobacteria, which is our blue-green algae. And then we've also got nitrogen fixing bacteria. It's super important. And then in food production, here's where we get to the froyo. So here's how yogurt is made. And it includes the introduction, well, first the removal of bacteria and then the introduction of a specific type.
I'm sure you know that yogurt comes from milk, but did you know that bacteria are also a key ingredient? Yes, bacteria, but don't worry, these are good bacteria and are often called probiotics. Your gut has thousands of types of bacteria happily living inside, whose job it is to aid digestion. Yogurt is thought to be a good vehicle to bring more of these good bacteria in. So in this video, we're going to learn a bit more about how yogurt is made. First, we need milk. Before we start the next steps, all of our equipment needs to be sterilized to kill off any unwanted bacteria and other microorganisms. Now we are ready to start. The milk is heated to somewhere between 85 and 95 degrees Celsius for between 15 and 30 minutes. This is a process called pasteurization and is also done to kill off any bacteria and other microorganisms that are naturally found in the milk. The milk is then homogenized. This breaks down the fat droplets in milk to make them smaller so that they stay suspended in the yogurt, rather than sinking and making it all lumpy. So we've sterilized our equipment, then pasteurized and homogenized the milk. Now the mixture is cooled to 40 to 45 degrees Celsius, and we add our special yogurt making bacteria. The mixture needs to be cooled first so that the bacteria aren't killed by the high temperature. You might be wondering why on earth are we adding bacteria when we've gone to all the effort so far of killing all the bacteria. We're adding lactic acid bacteria, which are also used in cheese manufacturing. The two species used to make yogurt are lactobacilli and streptococci. But don't worry, you don't need to learn these names. Just remember that lactic acid bacteria are added. These lactic acid bacteria are known as the starter culture. The bacteria ferment the milk and change it into yogurt. Lactose is the main sugar in milk, and during fermentation, the bacteria turn the lactose into lactic acid. The bacteria also start to digest the milk proteins. Fermentation takes many hours, and during this time, the mixture is kept at the optimum temperature of 40 to 45 degrees Celsius, so that the bacteria can work its fastest. As fermentation produces lactic acid, type of acid, the pH of the milk drops to about pH 4.4. This is why natural yogurt has a sharp, tangy taste. As the pH levels drop, the milk solidifies to become raw yogurt. Once the pH drops to a low, the lactic acid bacteria that make the yogurt also stop working, so the yogurt doesn't become too solid. The pH drop also stops any bad bacteria growing. The solidification happens because the proteins begin to coagulate. Coagulation is when something which is liquid turns into a more solid state. So we've sterilized our equipment, then pasteurized and homogenized the milk. Add lactic acid bacteria so fermentation happens, and then coagulation. The mixture is now stirred and cooled to 5 degrees Celsius, and we've made natural unflavored yogurt. At this stage, we can add flavorings and fruit. Now it's ready to be packaged and sold. Delicious. Can you match the words to the definition?
Delicious. And if you guys ever pay attention to what I'm drinking, as if you care, but I'm almost always drinking a kombucha in the morning. So you may catch me with kombucha when you're here in class. And it even says on the front of this is another thing where they use probiotics, right? Or your good bacteria. But it says 9 billion on the front of that, 9 billion living probiotics. And then back, it lists all the kinds they're using in there. So let me get in there. So leave that for a second there in case you guys want to pause and check answers on that section. All right, so this one too, which I forgot there was one more page. And you can check your answers. So let's talk about infectious diseases now. Diseases caused by viruses and prokaryotes. Okay, this is where it gets fun. So we have infectious diseases and disorders caused by organisms such as viruses, bacteria, fungi, or parasites. Many organisms live in and on our bodies, and they're normally harmless or helpful, but under certain conditions, they can cause disease. So here's this word again, pathogen. I told you we'd come back to this word. So in biology, a pathogen, which comes from the Greek word pathos and genes, is anything that can cause a disease, an infectious agent. And so pathos means suffering or passion, and not passion like I love you, but if you're a churchy person, passion like Christ's passion, Christ's suffering, same use of the word passion here. And then genus, which you would probably recognize as genesis or generate, meaning to produce. So it's anything that produces suffering. There you go. And a pathogen may also be referred to as an infectious agent, or you could say a germ. So it came into use, this term came into use in the 1880s. And typically the term is used to describe an infectious microorganism or agent, such as a virus, bacterium, protozoan, which we'll talk about next week, prion, which we're going to talk about today, viroid, which is really just kind of like the nucleic acid portion of a virus and not the rest of it, or fungus. So our human pathogens include these main categories here: virus, bacteria, fungi, protozoa, parasites, and prions. Prions you probably never heard of. If you have, then you're even nerdier than me, and that's awesome because I love these things. They're fascinating. So we're going to talk about them a little bit later. Pathogens, this is what they do. They can destroy your cells, damage your cells, or release toxins. Toxins is sort of a broadly defined word. It could mean they are releasing chemicals that damage the cell, which is kind of goes back to damage the cell. Poison, basically, anything that would be considered a waste product in the cell or dangerous to the cell. So we have what are called standard precautions for infection control, and it's capitalized because it's a thing, standard precautions or standard procedures. And in the past, this would have been a class that you would have sat through and gone, I might use those if I work in a lab or in the medical field, but some of this is kind of ubiquitous to what we're doing today. So hand hygiene obviously is going to be one of the biggest, if not the biggest and most important standard precaution for infection control: washing your hands. And like I said earlier, if you're using an alcohol-based hand sanitizer, you're breaking down either cell walls on bacterial pathogens or protein envelopes on viral pathogens, and that is helping make them either dead or ineffective, depending on what you're talking about. Use of personal protective equipment, PPE. Nobody would have known what PPE is 5 years ago either, unless you were working in the medical field. But yeah, PPE would be your personal protective equipment. So that's gloves, masks, and eyewear. Now that's ubiquitous. Respiratory hygiene and cough etiquette. They used to just say cover your cough. Now we're all reminded to cough into our elbows so that you're not getting it on your hands, because that goes back to poor hand hygiene. Sharps safety, so engineering and work practice controls. Sharp safety deals with needles and other sharp objects typically used in the medical field. So if you have ever noticed, like after a shot at the doctor's office, they'll stick the used syringe into like this little plastic box, a little flippy lid, so that nobody gets poked by it. If you work in surgical fields or anything where you actually are coming in contact, even just nursing, if you're coming in contact with sharps, and you get poked with a sharp, like a needle that has already poked someone else, then you have to go through a whole process. I mean, there's reporting involved and everything, because obviously there can be bloodborne pathogens that can live on that needle and get transferred from person to person. And then safe injection practices, aseptic technique for parenteral medications. Okay, so aseptic would mean clean, not dirty, clean it up, not sterile, but aseptic, as clean as you can get it. And then parenteral means a medication that is taken not through the digestive system at one end or the other. So not a pill or a suspension or a suppository or something like that, but rather a shot or an IV medication. Those would be parenteral. So viral diseases. Our good old friends these days. We've become very familiar, or a little more familiar than we maybe would have been in the past. So a viral infection is a proliferation of a harmful virus inside your body. Proliferating, it's going everywhere. They cannot reproduce without a host. So again, you can put bacteria down, and in a favorable situation, it'll just multiply and produce. But a virus is not going to do it. It can sit there on a surface and still be dangerous to you, and you can then pick it up and put it into your body, but it's not going to reproduce unless it has a host. So viruses infect hosts by injecting their genetic material, hijacking the cell's internal machinery, making more virus. And an active viral infection, when this happens, a virus makes copies of itself, and it bursts the host cell, killing it, to set off newly formed viral particles. And now they're free to go find another cell. And there are some cases where you get these viral particles that bud off the host cell over a period of time before killing it. But typically, we're talking more about them just exploding, whether it's lysogenic or a lytic cycle. Eventually, that's what's going to happen. So either way, they're free to go inject other cells. And symptoms of viral illnesses occur as a result of cell damage, tissue destruction, and the associated immune response. This is important right here, because this is what we've noticed with COVID. We've noticed in particular the types of tissues being targeted by specific variants is not necessarily the same, especially when it comes to Omicron. If you read an article, I shared an article with you guys to the forum like the day or two after you left for break. I'm actually going to make an extra credit assignment out of that for you all if you want. But they've noticed that Omicron seems to be more effective at replicating in particular types of body tissues rather than others. And that can potentially change the course of some things for us, which may be good. So the other thing we've noticed about immune response when it comes to COVID in particular is that it is often the immune response that is super dangerous. And this has been something that's been revealed over time with when comparing infections to people who are on, like, for example, biologic drugs. So I'm on biologic drugs for lupus, and those drugs suppress my immune response so my body doesn't attack itself. But it also suppresses immune response to other things like infections or pathogens that get into your body. So for a lot of people, what happens with COVID, at least with the wild type, the initial COVID-19, before it was mutated into a million variants, was people were getting it, and then their immune response was so overwhelming that they ended up being hospitalized because their own bodies were just like fighting really, really hard. The tissue destruction, some of that is immune response, and some of that is from the virus itself. But it was getting into like mast cell storms and all this other complicated business. So certain viruses, like the ones that cause chickenpox and cold sores, can become inactive or latent after the initial infection. So with cold sores, like you can get a cold sore and it'll erupt and then it'll heal. And then it just lays dormant in your body until a particular trigger comes along. And that can be stress, sunlight, all kinds of things. And then it makes that virus sort of reactivate, and it'll start reproducing again. With chickenpox in particular, what you get is a latent infection that can last for decades. So it can live in your nervous system. And this is varicella, chickenpox is varicella, and it can live in your nervous system for decades. And when you get older, what happens is some people get shingles, if you've heard of shingles. And basically shingles is like the reactivation of the chickenpox virus, the varicella virus that they probably caught as a child. And, now they could be a senior citizen with shingles, which is extremely painful because the virus, like I said, it's in your nervous system. It's in your nerves themselves, and it causes a lot of pain. So now they're doing, they have a shingles shot, a shingles booster that they can give to typically older adults. Most of you guys have probably had, or at least all of you are born within the time frame that the varicella vaccine has been available, so that chickenpox wasn't something that you personally endured. For most of you, probably. My generation, we had it. You wanted to get it because if you got it younger, your body seemed to fight it with less overwhelming immune response. And so, people used to do crazy things like have chickenpox parties. The kid down the street had chickenpox, and all the parents would like bring their kids over there so everybody would get the chickenpox, because you just wanted everybody to get it over with at the same time. But chickenpox can be very dangerous. I mean, we all thought of it as, it's just a thing. It can cause encephalopathy, which is inflammation around your brain. And actually, my cousin and I got chickenpox from each other, and mine was super mild. I barely had like anything. She had them inside her mouth, inside her eyelids, and then she ended up getting a staph infection, which is bacterial infection, secondary to the chickenpox because she had open sores everywhere. It was terrible. So be grateful if you haven't had chickenpox. Not fun. So vaccines, so how do vaccines work? So a pathogen, remember, is an organism that can cause disease within a body. And each pathogen is made up of several subparts that are unique to that pathogen. And we're going to use an analogy here of locks and keys, and I'm going to show you a video that explains it as well. But the subpart of the pathogen that causes the formation of antibodies, which is your body's response, is called an antigen. And if you think about a pathogen, just picture it like a little sphere. Okay, on the outside of the pathogen are these antigens, and they stick up. Now, go back to that image of that coronavirus I showed you at the beginning with all those little spike proteins on it. Okay, you can think of those like keys, okay, and they're all unique to that particular species of virus, okay? And then what your body does is it has sort of like soldiers within your system, and that you're fighting, right? That's your immune response, your fighting cells. But you've got particular white blood cells that'll go out and they're always looking for something, scouts. They're always looking for something that doesn't belong. And if they recognize something as new, they will recruit your body's locksmiths, and that's your immune system. And it's going to produce what are called antibodies, which are like locks that are specific to the keys or the antigens on that particular virus in this case. So when your human body is exposed to an antigen for the first time, it takes the immune response some time, and that's when you're ill. That's the time that you're susceptible to being ill. But the next time you come in contact with it, your body recognizes it. So once these specific antibodies are produced, they can destroy the pathogen, stop the disease. And antibodies to one pathogen don't usually protect against another unless they're really similar, which is, if you go back to that article that I shared with you guys, you'll see why I have some hypotheses I would love answered. Would love to see the answers. So love to test them. Once the body produces antibodies, it's primary response to an antigen. It also creates antibody producing memory cells. So these guys now go floating around in your body every day, all the time, and they are looking for anything they recognize. So if they recognize a particular virus, they go, oh, I know who you are. Hey, locksmith, send out the locks for this guy. And they will come and basically shut it down. So this means that a person exposed to a dangerous pathogen in the future, their immune system will respond immediately, protecting against disease. So what vaccines do is they expose you to the antigen without typically the exposing you to the disease itself. So your body can create the antibodies to the antigen without the effects of the pathogen. So here's your analogy, your lock and key. And this is a shorter little video, but does a good job describing what we were just saying.
Pathogens are foreign infectious microbes that cause sickness and disease. The job of our immune system is to fight pathogens. White blood cells are able to recognize foreign pathogens by their surface molecules. The immune system constantly scans the body for foreign molecules. If foreign molecules are recognized, the immune system will launch an attack. Foreign molecules that trigger an immune response are called antigens. The surface of a pathogen is covered in foreign antigens. Antigens have very specific shapes, and different types of pathogens have different antigens on their surface. When the immune system detects antigens, white blood cells respond by producing antibodies. Antibodies are special Y-shaped proteins that are made with a specific shape to match the antigen detected. Antibodies are marking flags that latch onto antigens just like a lock and key. Pathogens that have been flagged by antibodies are then destroyed by special white blood cells. The antibodies you've produced to flag one type of pathogen would not be effective against another. For example, the antibodies that flag the measles virus would not be able to flag the chickenpox virus. This is because antibodies and antigens work like a lock and key. The shapes must match. As you fight different viruses throughout your lifetime, you develop the ability to produce different types of antibodies. This is how you gain immunity against certain pathogens. This is also what vaccines do. They trigger your white blood cells to produce antibodies so that you become immune to a particular pathogen. Now you might be wondering, why do I get a cold virus every year if my immune system has already produced antibodies? Or I've gotten the flu shot, so why did I still get the flu? Well, some viruses, like the ones that cause the common cold and the flu, have many different strains. These viruses mutate frequently and develop different antigens. If the antigens change, then you will need to produce different antibodies to fight it. The flu shot will help you to produce antibodies against a few different strains of the flu, but there's always the chance that a different strain could infect you.
So we're talking about variants of coronavirus. You have to think back to those antigens on the surface. How many of those are the same? How many when it mutated? Because that's what's happening. We're watching SARS-CoV-19 mutate and evolve. We're watching evolution occur in live time, relatively short period of time, because it's a much more simple organism, right? It's not like a whole mammal or something. And so we're watching that evolution occur right now. And as it occurs, it changes. So now you have to wonder whether or not contact with one variant will be enough for your body to develop the antibodies to combat another variant. And we don't necessarily know the answers right now. In some cases, maybe yes. In some cases, maybe no. Oh, no, I already saw you. So viral infections, these are the most common viral infections in humans, just in case you were wondering. So, you can get stuff like measles. You've probably heard of that one. Oh, rabies is on the list. I don't want that one. Eye infection, you can get herpes in your eyes. That's the same thing that causes cold sores. It's herpes simplex. Rhinovirus, that one, that's your common cold. Epstein-Barr, you may have heard of. That's really not good. Mumps, you don't want the mumps. That's why we have a vaccine against mumps. Look right here, SARS coronavirus that can cause pneumonia. I actually have a friend just got out of the hospital for post-COVID pneumonia. And then you've got like Coxsackie, all the hepatitis. Here's your varicella. Varicella is your chickenpox. And then smallpox, which we've pretty much eradicated, thank goodness. Human papillomavirus actually can cause warts. And you'll see that it's here too, because it can cause warts in different places. And this is the one that is responsible for creating cervical cancer, actually, in women. And then you've got things like rotavirus and norovirus. These are all the ones that cause like the stomach flu. That's what you're talking about. So there are all types of viruses that infect all different parts of your human body. Leave that up for a second if you want to pause and then check your answers. All right, so let's now talk about some bacterial diseases. All righty, bacterial diseases. So bacterial diseases include any type of illness caused by bacteria. Normally, so you got millions of bacteria that live on your skin, in your mouth, in your intestines, in and on your genitalia, in your armpits. They're supposed to be there. They're part of your normal functioning. You want them there. So they're good bacteria for the most part. Everything that lives on you is going to be good unless you're sick. So sometimes we call them good bacteria, normal flora, or healthy bacteria. So your harmful bacteria that cause bacterial infections are the ones we call pathogenic. So we're back to this word again, right? And this happens, these pathogenic diseases occur when these pathogenic bacteria start reproducing, and they can crowd out your healthy bacteria that you need for normal functioning. Or they can grow on tissues that are normally sterile. There are some tissues that don't typically have any bacteria in them, harmful, good, either. So you don't want any bacteria growing there. And then your harmful bacteria also emit, there's this word again, this toxins, this loosely used word, that can damage the body. So what are antibiotics? You've probably had them at one point in your life. Most people have. So there are several classes of antibiotics, and they have completely different bacterial targets or act on the same target, but at a different place. So in principle, you've got three main targets for antibiotics. So antibiotics, if you look at the word anti, you know what that is, right? Against, no, bio, killing living things. So we're killing bacteria, not viruses. They don't work on viruses. So they're going to work on either your cell walls or cell membranes that surround your bacterial cells, the machinery that make up the nucleic acids, DNA and RNA, or the machinery that produce proteins, so the ribosomes in particular, because we know proteins are produced in ribosomes. We learned that last unit. So these targets are absent or different in the cells of humans and other mammals. So it means that when you take an antibiotic, it doesn't harm our cells because the mechanisms in them are specific to one or more of these three targets in bacterial cells. And they can have harmful side effects sometimes, or unpleasant side effects. And that isn't because they're damaging your cells, but rather because sometimes they're killing off the good bacteria. So if you've ever had a bacterial infection and they give you an antibiotic and you're like, oh, yay, I'm going to feel so much better. And then you get terrible diarrhea because it killed all the good cells, the good bacterial cells that were in your gut that are normally there. So that's why we have to, one of the reasons we should be limiting our antibiotic use. So they can have, there's either broad or narrow spectrum for antibiotics. So your narrow spectrum antibiotics work against more specific strains of bacteria, and your broad spectrum kind of kill much wider range. So you can think about it as like a sniper versus cannon fire. So if you want to kill one guy, this one guy causing you a problem, and you have a sniper, you can take out the one guy. That's your narrow spectrum. If you want to take out the one guy with your broad spectrum, you're going to blow up everybody around him too. Right. So the thing is, broad spectrum antibiotics are often used, and that's because sometimes it's hard for a doctor to diagnose exactly which bacteria is causing a problem. And they know that they're more likely to end the problem if they just prescribe the broad spectrum antibiotic, because it'll likely take out anything that's causing the problem. But sometimes you get a very persistent infection, and then they will culture the infection and see what bacteria is growing. So that's when you really would want to use a narrow spectrum. A good example of that, I have a friend whose son was helping clean a yard for somebody that he's close to, like an adult mentor. And a ball rolled down a hill, and he chased, and he was like 12 or 13 at the time, so not like a little guy, but he chased the ball down the hill and slid into a ditch full of muddy water and like grass clippings and all this yuck. But when he went in, his foot got impaled on a piece of rebar that at first they thought was a stick, but it wasn't, it was rebar. So if you ever seen rebar, it's like a metal rod. They use them like inside of the concrete in roads and stuff, to kind of hold things together in building construction. Don't know why it was there, but anyway, his foot gets impaled on it. So this guy calls his mom, who's my friend, and she's a doctor. So she goes over there, and she had to physically pull him off, which I'm sure felt fabulous. Took him back to her clinic and cleaned it up as best she could. And then texted the pictures to some friends, and they're like, you really should let a surgeon look at that, really get in there and clean it out. So she's like, ugh. So they go and they clean it out as best they can, they dress it, but it got infected, and he was on antibiotics, and nothing was killing it. So they got a little swab of it, they grew it in a dish, which is what this week's lab is about actually. And they cultured it, and they found out that the type of bacteria that was causing his infection is found typically among like leafy green watery plants. Go figure, it was living in the ditch. So it's not something they would have been looking for attacking a human body otherwise. So then they knew how to treat it. So that was a good example of when you would use a narrow spectrum. So your most common bacterial infections in humans would be things like, some of these you've heard of, I'm sure, strep, right? You may have heard of well, this is what causes tuberculosis. It's Mycobacterium tuberculosis. Staph, you've probably heard of staph. People get staph. And then you've got eye infections, sinuses, again, more STD ones down here. These are the ones that at least if you get any of these, at least if you get them, you can kill them with antibiotics. Whereas, like I said, the viruses, they have to run their course, or some of them will just lie latent in you, right? Food poisoning, you've probably heard of salmonella, E. coli, things like this, fun stuff. So here, I'll leave this up for a second, let you pause, check your answers. Okay, so emerging diseases. This is interesting stuff. So viral diseases are extremely widespread infections caused by viruses, a type of microorganism, which we already know. There are many types of viruses that cause a wide variety of viral diseases. The most common type is your common cold, which can cause a viral infection of the upper respiratory tract, nose and throat. Your other common viral diseases include things like chickenpox, not as common anymore, flu, herpes, that's your cold sores and warts and things like that. HIV, fortunately becoming less and less common. In some parts of the world, it's still more common. HPV, I mean, herpes isn't warts, sorry, HPV is warts. Herpes is cold sores, HPV is warts. Infectious mononucleosis, ever heard of mono? Yep. Mumps, measles, rubella, shingles. Now, shingles, as I mentioned, is your chickenpox years later. Viral gastroenteritis, your stomach flu, viral hepatitis, viral meningitis. These things sound terrible. Viral pneumonia. Viral diseases are contagious, spread from person to person when a virus enters the body and begins to multiply. So here's how you spread them. You can spread them through breathing in airborne droplets contaminated with the virus. This is the one we're dealing with now, right? All the time, constantly. Eating food or drinking water contaminated with a virus. Having sexual contact with a person who is infected with a sexually transmitted virus. Indirect transmission from person to person by a virus host, such as a mosquito, tick, or field mouse. We had the mosquito issues with Zika virus a couple years ago with the, it was in the Rio, Brazil, the Olympics. That was a big deal. Touching surfaces or body fluids contaminated with the virus. So viral diseases result in a wide variety of symptoms that vary in character and severity depending on the type of viral infection and other factors, including a person's age, overall health. Common symptoms of viral diseases include flu-like symptoms and malaise. Malaise is when you just feel like dirt. That's just, I just don't feel good. So viral diseases are not treatable with antibiotics because they're not alive. So antibiotic treating life isn't going to affect your virus. Now, sometimes a doctor will give you an antibiotic if you've had a virus so long that like your immune system's down and now you're getting bacterial infections on top of it. But in general, this is why it is not good to just go, oh, I don't feel well, I'll take this leftover antibiotic in my medicine cabinet. Don't do that. We'll talk about why. So however, the most common viral diseases, a common cold and the flu, are what we call self-limiting. So that means that the viral infection causes illness for a period of time, it resolves, the symptoms disappear, your immune system has attacked it, your body recovers, and typically you've now got antibodies that you will hang on to. So the next time you come in contact with it, you don't have to start over from the beginning. So this is super interesting, I found. So according to the National Institute of Allergy and Infectious Diseases, emerging infectious diseases are commonly defined as outbreaks of previously unknown diseases, known diseases that are rapidly increasing in incidence or geographic range in the last 2 decades, persistence of infectious disease that cannot be controlled. So NIAID's pathogen priority list is periodically reviewed and subject to revision in conjunction with federal partners, including the U.S. Department of Homeland Security, which determines threats and assessments, and the Centers for Disease Control and Prevention, which is responsible for responding to emerging pathogen threats. So makes perfect sense here, CDC. So when we're talking about Homeland Security, we are talking about biogerms, bioweapons. That's exactly what we're talking about, biothreats. There's certainly been some discussion and some inference that maybe that was one of the plans for COVID. Like some people think COVID may have been bioengineered, or maybe it was intended to be a bioweapon and it got out early. There are certainly bioweapons that already exist. I mean, if you go down to Atlanta to the CDC, you're going to find some of these diseases I'm going to show in just a minute that are housed there, which we keep contained. But if they got out into the public intentionally, that would be a bioweapon, right? So we have them categorized. We have category A pathogens, okay? And these are agents, biological agents that pose the highest risk to national security and public health. And the reasons would be because they are easily transmitted, they result in high mortality, people die quickly from them, and they can cause public panic or social disruption. And they require special action for public health preparedness. So these are what we call category A, and here's some examples of category A priority pathogens. Anthrax, probably too long ago for you guys to remember, but there was like a whole anthrax scare where people in Congress kept getting envelopes with white powder that people were thinking, oh my gosh, anthrax, it's a big deal. Yersinia pestis, plague, right? This is black plague, Yersinia pestis, bubonic plague, all right? You can see botulism is in here, smallpox. These are things you don't want in the public for obvious reasons. Dengue fever, Ebola, yeah, we don't want that. So then you have your category B pathogens, and these are second highest priority. And they are moderately easily disseminated or contracted from person to person. They result in moderate morbidity rates and low mortality rates. So less people are dying, but people are still dying. Require significant enhancements for diagnostic capability. Okay, so now look here. So we've got these lists of different categories and types, but in here we'll see things like this is the specific E. coli that causes like awful, dangerous, can kill you diarrhea. Ricin, you may have heard of ricin before. If you haven't, that's a fun one to look up. Staph, typhus fever, you've probably heard of typhus before. All kinds of crazy stuff is included in here. Some of these are very specific, like West Nile virus. You may have heard of West Nile. It comes through mosquitoes, right? Yellow fever, Zika. There's Zika I mentioned just a little bit ago, right? So these are all your category B. Now, category C are the third highest, and they include emerging pathogens that could be, read that, could be engineered for mass dissemination in the future because they're available. You can easily produce and disseminate them, and the potential for high morbidity, mortality, lots of people dying, and major health impact. So these are the ones that could be engineered. And here's the list. And what's right here on the list? There it is. That's why people have made such accusations, have had such thoughts, because there it is right there. Severe acute respiratory syndrome associated coronavirus SARS-CoV. And that would be COVID-19 would be one in particular. Also on this list, all kinds of interesting things like tick-borne illnesses, which you wouldn't think would be as easy to disseminate, but I guess they could be. All sorts of things. I'll leave it up for a second so you can kind of look through them, because look at there, poliovirus, right? All right, so prions. I'm kind of a nerd for prions. I think these are really interesting. So prions are diseases. The word would be transmissible spongiform encephalopathies. Transmissible, meaning it can be contracted from one person, contracted by one person from another. Spongiform, this has to do with structure. Think of a sponge, okay? You'll see why in a minute. Encephalopathy has to deal with your nervous system in particular. Encephalitis is like an infection of the brain area. So these are a family of rare, progressive, neurodegenerative, degenerates your nervous system disorders that affect both humans and animals. They are distinguished by long incubation periods and characteristic spongiform changes associated with neuronal loss. Basically, they will turn your brain matter into something that looks like a sponge, and you will lose neurons. And a failure to induce inflammatory response. So your body's immune system doesn't do what it should do when something is attacking, because these guys are different. Prion is really just a protein. It's not even a virus. Some people think they are like the evolutionary predecessors to viruses. So the causative agents of TSEs are believed to be prions. The term prion refers to this abnormal pathogenic agent. It's transmissible, able to induce abnormal folding of a specific normal cellular protein found abundantly in the brain. So there are particular proteins in your brain, and this little bugger causes them to fold wrong. If you remember, we talked about protein production. We talked about this little peptide chain. Remember, all those little amino acids come together, make this big chain, and then it folds in a particular way. It causes them to fold wrong. So your abnormal folding of prion proteins leads to brain damage and characteristic signs and symptoms of the disease. They are typically rapidly progressive, and always fatal. So they can take years and years and years to start functioning, but once they start doing what they do, it can go very fast. So you can actually have contracted a prion disease decades before it affects you. So your animal prions are things like bovine spongiform encephalopathy, which you probably may have heard of as mad cow disease. Chronic wasting disease, scrapie, which is in sheep. Transmissible mink, okay, this is in minks. This one is in cats, and this one is in ungulates, are like hoofed animals in general. In humans, we have Creutzfeldt-Jakob disease and a variant of Creutzfeldt-Jakob disease. Gerst-Menstrosler-Schleicher syndrome, that's a fun one. And fatal familial insomnia. This one has sort of invaded the human genome, but only in very few cases. It's very specific and it does get passed. And then Kuru. Kuru, we have a little video about. Kuru is fascinating. And our understanding of things like mad cow and Kuru go hand in hand. And then this is where your variant of CJD comes in. So here's mad cow disease. If you've never heard of it, you're about to find out. It's a very interesting little history blurb, and it shows you what you shouldn't do when farming and agriculture.
If you were around in the 90s, and especially if you lived in the UK, you might remember all the panic about mad cow disease. It's a disorder that's killed thousands of cattle since the 80s and 90s, and because a version of it can also infect humans, a lot of people were really freaked out. Today, thanks to safer farming, it probably isn't anything you need to worry about. And even though scientists still don't know exactly how it works, we can say one thing for sure. Your steak is almost definitely probably not infected. Mad cow disease is the common name for a condition called bovine spongiform encephalopathy, or BSE. It's a progressive neurological disease that affects cows, and over time it causes severe brain and nervous system damage, which eventually leads to trouble standing, walking, and changes in mood, like increased aggression and nervousness. The spongiform part of the name just means spongy, because the infection creates a bunch of holes in the cow's brain where its cells should be. The first 2 cases of BSE were identified in the UK in 1986, but it takes a really long time for symptoms to show up. So scientists think the first infections probably date back to the 70s. During the outbreak's peak in 1993, almost 1,000 new cattle were infected each week, but the number has gone down dramatically since then. BSE is caused by a bizarre self-replicating protein called a prion. Other pathogens like bacteria and viruses use DNA to make copies of themselves, but a prion is just a deformed version of a normal protein that's found in cell membranes. Sometimes those proteins can go rogue and get bent out of shape, but right now we don't totally understand how or why it happens. And when those proteins become prions, they can bind to other proteins like them and make them bend in the same way. And then those messed up prions corrupt even more proteins, and so on. Clumps of them collect and spread in the brain and nervous system, eventually causing brain damage. But that takes a while, so symptoms usually don't show up until years later. All of that is terrible, but how BSE got started is almost as horrifying. It probably happened because cattle were being fed ground up meat and bones from sheep and other cows. Ugh. There's another prion disease in sheep called scrapie, and it's possible that scrapie prions may have jumped to cows through their food and caused this whole mess. But it's also possible BSE just showed up when a random protein folded the wrong way. No matter how it started, the cattle feed only made things worse, because after cows died of BSE, they were ground up and fed to healthy cows, so the disease kept going. Unfortunately, a version of the disease can infect people too. The human version of BSE is called variant Creutzfeldt-Jakob disease, or VCJD, and it's also caused by bent prions. We don't know for sure that it comes from eating infected cattle, but since they're both prion diseases and both outbreaks happened around the same time, most scientists think that's the case. Like with mad cow disease, the symptoms of VCJD can take years to show up, but once they do, things move pretty quickly. Brain degeneration happens in just a few months, with symptoms like trembling, dementia, trouble walking, and eventually a coma. Since no cure exists yet, patients usually die within a year. Worldwide, there have been about 230 cases of VCJD, and about 180 of those were in the UK. The rest were mainly in Europe, and there have been only 4 cases in the United States, but they were all picked up overseas. The rate at which people have been getting infected has gone way down since the 90s, but scientists will keep studying it because there are other kinds of Creutzfeldt-Jakob disease not caused by cows. Before mad cow disease was a thing, we knew about CJD as a rare condition that could appear if a random protein went bad, usually through an inherited mutation or through medical procedures like transplants. Today, we keep the risk of very variant CJD low by giving cows safer food and making sure no nervous system tissue gets into our beef. And even though the risk is small, you also can't donate blood in the US if you spent too much time in high-risk countries or got a blood transfusion in Europe, because there's a chance prions could be spread through blood too. Mainly it's those new farming practices that have really helped get the disease under control. And in 2016, there were no new reports of BSE in cows in the UK for the first time since the outbreak started, which is both exciting and a relief. Also, because mad cow disease is transmitted through nervous system tissue, there's no evidence you can get it from milk or the meat used to make things like hamburgers and steak. So your roast beef is not out to get you, which is always good to know. Thanks for watching this episode of SciShow, and special thanks to Patreon patron Scott Sorrell for asking us.
Thanks, Scott. So, yeah, that does say cannibalism. We'll get there. So with mad cow disease in particular, it was very likely caused by feeding cows ground up sheep, because sheep had a already known prion in their population called scrapie, which called scrapie because they rub their heads on fences like incessantly. They can't stop. One of the things they do. But cows are herbivores. They're not supposed to eat sheep. But if that's all you're giving them is ground up bones and brains and things of sheep, they're going to eat it because that's all they're giving them. And then they would grind up the other cows and have the cows eat cows. And then, oh, well, that's cannibalism in a way, right? Cows ate cows. And then before you know it, all these cows have this neurological issue. This is also something to keep in mind. He mentioned like farming practices and making sure that no brain matter gets into or any nervous tissue gets into the beef supply. So the reason you can eat a steak rare, like if you like, I like my steak, I like a good medium rare ribeye, is because it's all enclosed, right? Like the only surface area is what's on the outside of the steak. All that other muscle tissue is contained. But if you have ground beef, for example, like a hamburger, the reason it's smart to cook that all the way is because all of the ground beef came from various other parts of the various other muscle tissue that's all ground up. And sometimes there is not good practice within a slaughterhouse and something will hit the floor or whatever, and they'll pick it up. And maybe the floor also has brain and nervous tissue on it, and you could get prions passed that way, or it could have more commonly E. coli from the cow's waste on the floor, and then that gets picked up and into your hamburger. So this is why you can have a rare steak, but maybe want to think about it with your hamburger. We've gotten much better at this, but still. So this goes back to what I said was super interesting, kuru. Okay, kuru is a prion that affected humans, and this has always been a fascinating thing to me. So again, kuru, mad cow disease, Creutzfeldt-Jakob, the three pieces together sort of helps complete the puzzle.
In the 1930s, Australian prospectors went to Papua New Guinea in search of gold. There they encountered tribes that no one had seen before. One of these tribes was called the Foray. In the 1950s, scientists came to the area to study these people and made a disturbing discovery. Out of the total population of the Foray, numbering about 11,000, roughly 200 people died each year from an unknown disease. Patients at first had difficulty walking and speaking, then began to suffer tremors, and in the later stages of the disease, lost all muscle control. They couldn't get up, they couldn't feed, and inevitably, they would die. The Foray called this disease kuru, which means trembling, and it's the name by which it became known in medical literature. A curious thing about it was that almost all the patients were women and children. Scientists searched for years to uncover the cause of the mysterious affliction, and finally they found it: cannibalism. The Foray cooked and ate their dead, and they did it as an act of kindness. The Foray believed that it was much better for a body to be eaten by people who loved the deceased than by worms and insects. Women and children ate the brains of the dead, while men ate other body parts. The most interesting aspect of the disease, it turned out, was that the pathogen which causes it isn't a virus, a bacterium, a fungus, or a parasite. It's a type of infectious agent that's completely new to science, one that could survive even at boiling temperatures. This newfound pathogen was a protein called a prion. Prions are proteins often found on the surface of our cells, but if we eat infected meat, eventually the protein reaches the brain and its structure changes and becomes misfolded. The effect of the misfolded protein is to cause neurological damage and ultimately death. After this discovery was made, the foray stopped their human-eating ceremonies. Although it's difficult to be sure that Kuru has completely disappeared due to the long incubation period of between 10 and 50 years, researchers think that the last Kuru patient died in 2009. In 2012, the epidemic was officially considered to have ended.
So that's what happens when you eat brain matter that's infected. It's the same thing that happened to the cows that ate the cows. So it's interesting because that was a completely different cultural experience than any of us have probably ever had. And that particular group of people was doing what they felt was the respectful thing to do. And that was their tradition and their culture. And they did not realize that it was making them incredibly ill. So there you go. That's Kuru. Fascinating. I think Kuru is fascinating. So anyway, you have to end on cannibalism. So lab 18, here's what we're going to do for lab 18. I'm going to put out a lab video, most likely by Friday. I'll send an announcement once it's out. I can't do all of it yet because this was the issue with more supply chain stuff I ordered in July is going to now be late. But it should be here by tomorrow. I've got a tracking number now. And so you wouldn't have been able to do it today anyway, but I had to do something else. So I will get it filmed for you and I will put that video up. And then also attached to the assignment will be the video. And then there is an analyze and conclude answer sheet that you can download and print and then complete it and bring it back to class. It shouldn't take you but 10-15 minutes to complete the answer sheet once you've watched the lab video. And if for some reason the video production and publication gets delayed, then I'll push back that due date. So I think that's it for us today. Does anybody have questions for me or are you guys all good? Let me know if you got a question. Otherwise, I think y'all are good to go. All right, so we will meet next week like normal. I hope. I hope that's where we're going. Should be. I don't have any reason to think we're not. So we'll meet next week and we'll move on with our next lab and all of our next stuff. And if you have a question between now and then, let me know. Your email will come out shortly as soon as I get this video link up. And you need me between then and now, you know where to find me. Okay? Happy New Year. See y'all later. Bye.